folks a chance to join for the meetings that end at 11 if you're on the East Coast. Okay, good morning and welcome to the Star Trek High Fidelity Seminar Series. It's a great pleasure to introduce today's speaker, Dr. Thomas T.C. Chung, who is the Director of Translational Programs for the Conrad Prebys Center for Chemical Genomics at the Sanford Burnham Prebys Medical Discovery Institute, where he leads strategic collaborations with academic and clinical translational centers to focus on translating basic research into new therapeutics. He co-developed and manages a strategic alliance in drug discovery with the Mayo Clinic and the University of Southern California. He also co-developed and managed the Collaboration for Cure program, a local San Diego initiative to seed translational research towards cures for Alzheimer's disease. TC earned a Bachelor of Science in Chemistry from the Massachusetts Institute of Technology, did graduate work at Stanford University, and was an Industrial Affiliates Fellow in Biosciences at Exxon Research and Engineering before earning his Ph.D. in Chemistry from the University of California, Berkeley. TC has over 25 years of experience in drug discovery and accomplishments in the departments of medicinal chemistry and molecular pharmacology at SmithKline and French Research Laboratories, which is presently GlaxoSmithKline, in virology with Merck and Bristol Myers Squibb, and high-throughput screening biology at Pharmacopoeia. He served as the Senior Director of Lead Discovery and High-Throughput Screening at DuPont Pharmaceuticals and as the Chief Scientific Officer of Genomics. He also helped establish the Office of Translation to Practice during his tenure at the Mayo Clinic. TC serves as a science advisor, business and technical consultant to life science, pharmaceutical, diagnostic, medical device, and specialty chemical companies. He is also the President Emeritus of the Society for Biomolecular Sciences and an Associate Editor of the NIH NCATS Assay Guidance Manual. While at DuPont Merck, TC's team originated the Z-factor, which is the most cited performance reference metric in assay development and high-throughput screening. Today, he'll tell us the story behind the Z-factor. Welcome, TC.
Well, thank you, Nathan, for that really generous introduction. So as you can see for the logos that I have on the bottom of my slide, that's what Nathan just went through it. I actually spanned a 16-year career in commercial pharmaceutical and biotechnology companies. And surprisingly, I found myself just entering my 16th, finishing my 16th year here in an academic drug discovery center. So during that entire time, I've been exposed to assays, the need for assay development, and the performance and needing robust assays, as I was responsible for many high throughput screening assays and campaigns. So I wanted to kind of give you a little bit of the story behind what how the Z-factor came to be, and some of the discussions that led up to that. And all of that, really, the seminal period was during that time that I have boxed at my time at DuPont Pharmaceuticals. In particular, the genesis and the discussions that sealed that really happened at the 5th annual conference of the Society for Biomedical Screening in San Diego, when at the time I was living in Delaware. So just as an outline of my talk here, I wanted to just start with, some HTS assay considerations and challenges, and how random error plays a factor in that. Then move on to the genesis of this idea of a simple parameter that can reflect our intuitions of what a good assay is. And then, go through the mathematical and graphical formalisms for what we call the Z-factor, walk you through some of the Z-factor ranges, as it relates to expected assay doability and quality, illustrates some of the HTS data and the relation to Z-factor components, and how we can use the Z-factor for, in addition to assay QC, how we can actually use it during operation of the screen. Some thoughts about hit confirmation and what considerations lead into that, what one can expect for rates of hit confirmation. And the Z-factor story hasn't stopped. There's actually been a lot of response more recently to some of the weaknesses or failings of the Z-factor, and I like to bring that up and put that in perspective. And just conclude with some thoughts about what I think an effective screen is, because we're often asked to rescreen libraries and things of that sort, and just end with some concluding thoughts. So with that, just, to begin, before we even talk about, assay robustness, let's talk about how we make assays robust in terms of making sure that, we have the right considerations. So, all the assays that we're going to use in HTS and lead optimization support in drug discovery, they need to be validated for even before assay and noise. We need to make sure that the assays are really well matched to the biological and pharmacological needs and relevance to the target and the disease. You want to have control over the reagent stability, so you want to check the reagents are stable during the assay, during the performance period, during storage, and that it's stable to freeze thaw. And if it's not, you need to aliquot them and check them for long-term stability for temperature, and keep working stocks, and whether you can pre-mix samples and use them, or do you have to keep them all separate, and whether you can use leftovers after you refreeze them. The other thing with getting an assay robust and improving its performance is really try to eliminate all the systematic errors that one can see. So you have to check the assays. These are all known for most of us. You check the DMS compatibility. And we are constrained to do many of our assays in these microtiter plates. And so each plate has its own characteristics for thickness and manufacturing uniformity. So we want to make sure that when we actually do an assay, we can check to see if there's edge effects. Because the edges of wells are closer to heat sources, and they actually are more affected by variations in heat, whether or not you have inadequate humidity, so things dry out. And you can notice things like striping during assay performance, and you can go through ways you can check on that. You want to make sure that from plate to plate and between day to day, that the assay performance is pretty reasonable. And there's standard things like looking at max and minimum values, some of the standard deviations, and signal to background and signal noise, which we'll go after in a little bit. And we also want to make sure that our assay is in the linear range. If it's a kinetic assay, for instance, that the signal is really the time signal versus time is in the linear range. So you want to do a time course so that you can actually make sure that you're not running out of reagents and getting the wrong number. You also want to check the response versus the target concentration and see if any of your reagents are limiting, and that's actually leading to a limitation of the signal and reducing your signal range. If you take care of all those systematic errors, then the only errors that remain are at random. The noise and the noise of the system and in the measurement. The other thing too, the other assumption we all make is that, and it's a fairly good assumption, is that most of the compounds in a screening deck are not active at the concentration we test them at, so that their activities really approximate noise. Noise is normally distributed, that's Gaussian. So this is all summarized in a very good chapter that Phil Iverson actually championed for factors for assay validation in the assay guidance manual here cited. So we all use microtiter plates. I just show this because the preferred standard in ultra-high throughput screening is, the 1536 ball format. And we're all familiar with all of these are basically multiples of the original 96 well, which is 8 by 12. And then the other intermediate plate is really for well plate, which is 16 by 24. And typically, in many libraries, the first couple columns are empty, and that's where you put your controls. The samples are actually all within the bulk of the main center. In our case, during our time at the NIH Molecular Libraries program, we actually received plates that were originally in 96 well, and then we had actual empty columns in columns 1 through 4 and the last 4 columns as well. So we actually had a total of over 200 controls. So we had a very good statistical representation for high controls and low controls. And we typically set up our assays so that the number of high controls were larger in replicates, because often that's where the signal variation is the greatest for the high values of whatever signal you have. I belabor this mostly because for 1536 plates, edge effects are most profound and exaggerated, especially when you have long incubations for live cell assays that might take up to 5 days. And we had to take heroic measures to actually reduce variation of temperature and humidity by using things like these commercial microclimate plates, which allow you to saturate the top lid with buffer in an absorbent membrane, or in these old Calypsis metal lids, which actually had a lot of thermal mass and restricted the access of CO2 to reduce the amount of evaporation that happened. And these worked. These are particularly heavy and needed special clamps, but they actually allowed us to do that. The other thing here, these tables I created really emphasize that the contacted surface area, that is when the volume is in that contained space, all the surface area that the liquid volume contacts is very high, proportionally very high in a 1536 well plate compared to a normal 96 well plate. What this means is it accentuates all the surface area effects. So it makes adsorption phenomena like ELISA's better, but it exacerbates compound loss if compounds tend to be sticky and stick to the plate. The other thing that most bulk dispensers in 1536 well plates actually go in a serpentine fashion. And that's all well and good, but if you have a block tip in a dispenser, you can actually have a striping where you have not enough reagent being added. So these are characteristic things to look for. And when you actually run plate-based assays, this was covered very well in these 2 references here with Phil Iverson in the Assay Guidance Manual and Kevorkis and Mark Orenkikurov in 1986, in 2005 with the JBS, to basically plot your screening data either across by well number and by row, or then by columns, or by a column view. But what we're looking for is basically smooth trends that bounce around, but on average do not show any effects. So that would be a good assay. But if you have edge effects where near the end of every row you have something happen systematically, that would be an edge effect. One way to check edge effects is also physically take the plate and flip it and see if all the values also translate with that rotation. The other thing that can happen is, instead of just pronounced edge effect, you can actually have things like for each plate and across a run, there's a systematic drift that goes from well to well, and that usually indicates that it's not an edge effect, but basically a gradient across the plate. So if you do these kinds of controls and you can kind of see that there's something wrong, you can correct your assay and so that you achieve something like this. So once you get to that point, you can start thinking about whether your assay looks robust or not. Now, the challenge of HGS assay is that we only test a compound once. So we really need a high degree of accuracy and sensitivity in the assay. And we want to make sure we can be confident that assay is robust, because it's critical to identifying hits. And so, as we just discussed, even if you remove all systematic errors, there'll still be, random errors from the instrument, just the measurement, and human-associated random errors. For instance, if you're measuring weights and you read the values, you might mistranscribe them. If you're looking at lengths or things of that sort, it's your eye that looks at the calibration. But intuitively, we know that the lower the measurement variability, then the higher that we're sure that the value we measure that's different from that background is a hit, so that's real, and that will ultimately confirm that upon retesting. So we've had traditional measures of assay variability, such as standard deviation and coefficient of variation, but these are actually insufficient to address the overall assay quality robustness, and I'll show you that in 2 slides hence what I mean by that. I put this slide in just to remind us all that, random error is normally distributed. So it's Gaussian. And the characteristics of a Gaussian distribution is that in this bell-shaped distribution, it's symmetrical across the actual mean, is that 97% of all the values fall within 3 standard deviations of that mean, so that things outside of that have a higher chance of being different than the background noise of the mean. So this is something we always take that comes from the elementary statistics here, it's cited. And I know we're all very familiar with that. So the way the idea of a z factor came in this way, and I'll define that more clearly in this minute. But if you look at these 2 graphs on the left, A and B, even without too much prodding, if you simply look at the scatter of a high signal and a low signal, and look at both of these, you can actually just intuitively feel that the top assay is a better assay in terms of discriminating whether a particular value belongs to the top population or the bottom population, or is different from either, right? If you look at the bottom, there's a lot more scatter in B, and the 2 bands seem to kind of interfere with each other. So the Z factor idea came because we noticed this visually intuitive feeling that panel A would give a more useful assay than panel B for calling hits. Now, there are 2 common parameters we're all very familiar with. One is signal to noise, and that's defined here where it's basically the mean signal divided by the mean background over the standard deviation of the background. And a signal to background, which is, the ratio of the mean signal to the mean background. When I started my career at Merck, I was told to have, Z signal to backgrounds that were just very large, 10-fold to 100-fold, and anything like that would make a good assay. The trouble is, either of these parameters by themselves really do not capture an assay's totality. It's either alone is poor at distinguishing because neither takes into full account both the variability and the separation between the dynamic range between those 2 values. And to put it in perspective, for this example from our paper that we published on the Z factor, you can see that the noise here is bracketed by the Gaussian distribution. So 3 standard deviations from the mean is what this line purports to be. And same thing for each of these, if we treat them as random errors. So these are the population distributions. For the top panel A, you can see that the signal to noise for this example is actually 12. You take the signal 50 divided by the noise here, 10. And you divide it by, standard deviation of that background, which is in each case 10 divided by 3, so 3 standard deviations. So you can see that while the top has a smaller signal to background compared to the panel B, you can still see that panel A would be a better assay. So the signal to background doesn't capture it. Signal to noise here is even lower than here. So again, even the signal noise is good. So neither of these alone captured our intuition that panel A is better. The Z factor, which I'll describe in a little bit, actually is the one parameter that captures that. So here the Z factor was larger than the Z factor for B. And it correlates with our intuition just looking at this, that it's a. So I want to emphasize that this is a simple mathematical formula that I'm going to present. The only statistics that we kind of appeal to is the idea that noise or the bandwidth of the signal, top and bottom, high and low signals are Gaussianly distributed. So that gives us the 3 standard deviations from the mean. That's our 3 standard deviation number. Before I show you the formula and go walk through it, I kind of just want to tell a little about the Z factor. You know, we had this conversation at DuPont Pharmaceuticals when I led the team, and it was first brought to my attention with Jihud Zhang, who formalized a lot of this and drafted some of the use cases for it, and that's why we named it the Z factor. It's actually Jihud Zhang, so this is Zhang factor. Now, our second in command, Seth Kevin Olenberg, championed the idea that this would really be a useful and highly cited metric. I thought it was very intuitively obvious and therefore somewhat trivial and kind of not worth publishing. However, later that year, you know, as I said, at the annual conference in San Diego, Kevin and I joined a dinner cruise hosted by LJ Biosystems, and there was a guy, Doug Moblin, who helped co-develop one of the LGA systems analysts machines. Is a good physicist and had a good idea of what he wanted. He was so enthusiastic and embraced the Z factor idea because it was simple and intuitive that it removed all my hesitations. So we then quickly published the Z factor paper. So had I prevailed, it could have been delayed for a while, but and the Google Scholar actually I just checked this recently, shows that, you know, there are almost 7,300 citations from in Google Scholar. Web of Science lists about, you know, 5,300 citations. There are over 100,000 just mentions on the web and stuff. So, you know, Kevin was right and I was wrong. So this is the reference. So let's walk through the definition of what a Z factor is and show you the graphical interpretation of that. So here's the formula. It's a very simple mathematical formula. All it is, it's really the normalized ratio of the sum of the 3 standard deviations from each individual signal or control, or sample or control in this formalism from our paper, divided by the absolute difference of the mean. So it's just a proportion, and you subtract that from 1. The way we interpret this is graphically is if you have a sample mean, it's going to have a distribution, and a control mean has a distribution. At 3 standard deviations from the mean, this is the sort of the variability zone for each random data. And you're simply asking the question, is there a separation between this envelope and this envelope? And here there's a clear separation. The Z factor is actually dimensionless and it's relatively easy to calculate. So note a couple of things. The Z factor is very highly sensitive to data variability because it's 3 times the standard deviations, that factor of 3. In our paper, we formalized this as talking about the mean of samples, but you can actually simply, for an assay without compounds, you can simply put a reference high control and a reference low control, DMSO if it's low value, and a reference compound if it's a high value. The formula is symmetric. You just have to define your controls to give you high or low values in a consistent way. The assumptions for this whole exercise is that these variabilities are normally distributed, and that's true for controls that, like DMSO, that have no compounds, or large unbiased compound sets like HD's compounds, because most of them are inactive. So you can formally also calculate a Z factor, we call it Z prime, if instead of samples, you just have controls, high and low controls. It's just simply a formalism. So let's go through some of the use cases for kind of talking about what ranges of Z values mean something. So if we set Z to 1, that's an ideal assay. it's ideal in the sense that it's an unattainable theoretical limit. The way it works is so here I summarize if Z factor is 1, it's ideally, and actually Z factors really can never exceed 1, because if you look at the formula, it can never be higher than that. What that means in terms of limits is you get it in two ways. One is the numerator here, the individual bands all go to 0. What it means then is you have a case in which the actual, no matter what the mean of the sample or control is, the distribution is just a straight line. There is absolutely no noise in either of these signals. So if that's true, then the separation zone is really infinite because this is 0. So separation zone is really infinite because there's no noise. So these, even if they come close to each other, there'll still be a separation zone. The other way that can happen is if the two means are widely separated as they approach infinity. That means these means are so well separated that it doesn't matter kind of what the data variability is. There's a huge and infinite separation zone. Again, this is a formalism and an extreme that can't be achieved in real life. So what's more useful is looking at when Z prime is between 1 and 0.5. And we choose this because when Z is greater than 0.5, you have this condition. Okay, but if you keep pushing these means together and reducing until you get to this stage where you actually have the numerator here equal to the denominator here, that is equivalent of having the signal sample and control variations equal to each other and doubled, so that we have 12 standard deviations of separation. So this is how we retrieve the value 0.5 being a good assay, from 0 to 0.5. So there's a clear separation zone. So this is what we term an excellent assay. This is the common metric where we say, you have an excellent assay if Z prime is greater than 0.5. Now that's all been overemphasized because you can look at the next situation is when between where Z is 0.5 to 0. What happens is we start with the Z of 0.5 where there's good separation, and we keep moving them closer and closer until the two regions of data variability touch. Upon when they touch, then you have no separation band. This green band disappears. So it's equally likely that that a value is in this envelope or this envelope. So therefore, it's really a coin toss. It's a yes/no assay, and that's how we get to that 0 being a coin toss assay. Not very useful for choosing hits, because it's always 50% one or the other. However, between 0.5 to 0, you can still have separations, and it's still an acceptable assay. It's doable. You can actually improve assays that are less than 0.5 by running replicates, because then the data variability will decrease as a function of the square root of n. So I emphasize this because I think many of the reactions to complaints about the Z factor, it just says that assays have to be greater than 0.5 to be good. That's not true. You can have doable assays lower. In fact, in our history, we've run assays with Z primes of 0.2 to 0.6. And I'll go over some of the complaints to that later. The reason why when Z prime is less than 0, it's unsuitable is because at that point, what you're really saying is the signal and the sample and background bands actually overlapped each other, so that not only do you have no zone of separation, the zone of separation becomes negative because they run over each other. So you can't choose whether a hit is either in this envelope or this envelope. So that's the factor. So when the Z is less than 0, that's what happens. It overlaps. In the extreme, if If the sample mean and the control mean are equal, this collapses to 0, and this value becomes infinity. So in theory, z prime can range from -infinity to 1. Yet practically in terms, negative z primes are meaningless because at that point, you're already saturating the entire system and you're at the lower limit of detection. So when z prime is -1, that's what you're at. So you can actually use, if you set the z2 -1, you can put in the formula and calculate that the sample mean must equal the control mean plus 3 standard deviations of the control mean. So that sets the lower detection limit, and that's how you can use z prime to calculate using reference extreme reference controls. So you can compare two instruments, see which is better than the other. So how does this look for, so this is an example of an assay in theory where you can look at the low control and the high control, and then calculate the z factor, z prime factor, just for the controls and how it relates to when you have the z factor for the actual screen, where instead of using this low control, you use the actual assay envelope itself. So it's the same formalism. What you should always realize is that the z prime is always going to be larger than the z for the assay because there's more noise and more spread in the full screen. So as as a frequency distribution, you can see that the separation band mirrors what we just did for the formalism. And for the a scattergram, you can see that it would be the separation bands. That's clear. So the advantage of the Z factor is it's really simple. It's an idea of a separation band, and you just have to remember that the ratio is going to be greater than 0.5 to be a good assay and above 0 for a doable assay. And it all reduces the variability and the dynamic range into one simple parameter. This is an example of an actual assay and the calculations for the Z prime and the Z. This is an alpha ELISA assay in which we've, sorry, it's a TR-FROD assay in which we've had streptavidin and it's looking to block this. And we have protein-protein interaction. In this case, the pilot screen was done with 7,000 compounds and 3D-MSO plates. The 3D-MSO plates were used to give us our Z prime. You can see that we calculated the Z prime and it was 0.72. And with the pilot number of compounds, we had a slight degradation in the Z factor. And you can see that when with the full screen, the Z of the controls were close, and the Z factor of the compounds also degraded. So again, whenever you run a screen like this, ideally what you want to see is the controls truly are Gaussian. So low controls are truly Gaussian. In this case, there was some skew in the higher controls. And then if you look at the envelope of the screening samples, again, it looks mostly Gaussian. There's a little tailing here. And you compare both the pilot and stuff. The other way you can use, and so this is the other view of that. The other way you can use the Z factor is doing the actual run of a high throughput screen. So in this case, we had sample plates through the entire run, and we calculated the standard, the signal to background, the signal to noise, and saw that they were fairly, good, as well as signal window. The Z factor that captures all of this actually was fairly consistent. In one case, we had a higher value, but it was pretty overall self-consistent. So this is how we kind of check how you can use Z factors during run to QC the plate variability and make sure the performance doesn't degrade with execution. So note on hit confirmation rules of thumb. So when we define a hit, we set a threshold, and the standard deviation of the compounds, 3 standard deviation mean sets that threshold often. Once you set a threshold, the question is, if you have a hit that's a certain number of standard deviations away from the threshold, what's your expectation that it would actually reproduce? The important point is if a compound you define as a hit is right at the threshold, no matter how precise the assay is, that compound will retest half the time. It will not be above threshold. So the reconfirmation rate of a compound that's right at threshold are always going to be 50%. And in this example here, if the mean is 5 and the standard deviation 15, we set a threshold at the mean plus 3 standard deviations, which equals 50. So if you have a compound that's at 3 standard deviations from that threshold, here, then you would expect that to confirm 99.7% of the time. But as you get closer to the threshold, the confirmation rate goes down in a Gaussian fashion. What practically this means is what it practically means is the hit confirmation rates better than 35 to 40% means you have a very good initial screen, and the confirmation is in acceptable range. If it falls below 20 to 25%, you should be concerned that there's something you're missing in the assay, despite its seeming to be robust, that there's some systematic thing you're not picking up. You're having a lot of false positives. So that's also true if you don't have a reference positive signal, high signal, which is often true when you're looking for an agonist and trying to deorphanize a receptor where you don't have a reference compound. So there you can only look at the distribution of the main envelope away from the mean. You don't actually know how far you are from that. It's just the Z factor, sorry, the Z score. So I talked about how the Z factor was very simple and intuitive. And again, it really wasn't statistically rigorous. Prior to the Z factor itself, there was a very useful metric from Sitalampalam et al., Sita's group at Lilly, where they published something called the signal window. And it was useful, but it was complicated because there was 2 different cases for the window size if it was an activator versus an inhibitor screen. So one had to remember a couple of things and put together. There was an alternative to more recent alternative to the HTSQA, where they try to remove the sensitivity to the effect, to the size of the distribution and number of samples, because there's an effect size, and that there is a relationship between the mean and variance. And the way their solution to that was to take a log transformation of the distribution and set up power calculations. So this is a kind of a useful variant of the Z factor to actually try to determine the power of an assay to actually call hits and to reduce false positives. So you can read that paper. A very useful adjustment to the Z factor is something called the robust Z prime, championed by Burnham et al. In 2019. And that's actually to take, to substitute for the mean, the median, and the median deviation for the standard deviation. And this was used to minimize the effects of extreme outliers in either the high or low controls, because, as I said, the Z factor is quite sensitive to data variation because of the 3 sigma factor. We use this in our group too if we have a noisy assay, and it's a very useful adjustment to simply substitute the median for the mean. More recently, in the SLA discovery, there's been a useful software that was described by Barr and Zweifach, where they were really they're focused on assays, using assays, and how to set thresholds and assay quality for where this formal Z prime is less than 0.5, the range where I talked about where you could do duplicates and replicates to make the signal go, the variation improve. What Barr and Zweifach found was that they confirm the intuition mathematically that, and the formalism that the Z factor, Z prime of greater than 0.5, assays that have that do in fact perform better than assays with Z factors less than 0.5. But they object to having a strict cutoff of Z greater than 0.5. Now, again, I point out that we never said in the original paper that it had to be this. We said that if it is this, it's a very excellent assay. But if it's lower than 0.5, it's not a doable assay. The value of Bar and Zweifach's formalisms and the way they define that is they actually calculated where you could actually have useful cutoffs and predict what the reproducible and confirmation rates would be. So they came up with these formalisms that talked about defining where defining a threshold where 80% of the compounds that you identify as active really do inhibit greater than 50%. So this T 80% value is what they calculate. And they have this formalism. You can see that the fraction of compounds they find as the Z factor goes up from 0.5, and even though it's below 0.5 here, there's still acceptable number of fractions. So, you know, it's not they're hitting less than half, they're more than half of them. So this is a useful adjustment to trying to find confidence of hits when Z factors are below 0.4 to 0. However, you do have to refer to the formalism and calculate these. It's much more complicated. Much more recently, there has been this paper from X.D. Zhang, not Jihu Zhang, who is an informatician and published in Bioinformatics. And he really spent a good time detailing 4 major issues that he had with the Z factor metric. Mostly to say that it's not a rigorous statistical, it's not statistically based, and it's misuse of some of the formalism statistics, that it violates Pythagorean theorem, that there's no adjustment for sampling error and effect size, and that really there isn't a good mathematical grounding to use 3 standard deviations from the mean statistically. So I show you all this, not so that I'm going to walk through everything, but really to show you the complexity of the thought process, and the math and formalism is complex. It all distills down to giving a new rule of thumb that talks about how you can actually rescue hits that you might miss if you kind of say a 0.4 Z prime or less assay is not, you know, suitable, or you ignore those assays. They're trying to basically recover hits and use assays that have lower Z scores and try to tell you how you can actually still identify hits with confidence. But you can see it's fairly complex. I've walked through this paper twice, and I'm still trying to understand the nuances. I think, however, this table 2 is the best summation of what you can use for it. Even with these guidances, you can see that you have to kind of really think through how you're going to use each of these, okay? So I kind of want to close by sharing something which I think about in terms of assays. So, you know, we can treat assays, high throughput assays in Gaussian fashion because most of the compounds are inactive. And often, so I show this because if I actually gave you the thought experiment of asking you to screen 300,000 compounds, but I actually, instead of giving you a random library, I gave you replicates of a single compound with an IC50 of 10 micromolar. If I screened it at a concentration of 10 micromolar with IC50 10 micromolar, then I should expect here 50% activity as my mean of my distribution of my population. And this data variability will represent basically the instrument noise for this IC50. It's basically replicating a Gaussian dose response curve. if I go very low, so saying a random compound is not active is like saying I take a true active compound and dilute it until it's not active under conditions. So this is what you'd expect. If you get to higher concentrations, you would actually change the mean of the activity value. But what you would do is you would actually tend to spread the noise, because as you higher concentration, we all know that you have, the assays become less robust to them. So I expect you would actually increase the noise. In a random library, most of it should act like inactive compounds. But if you look at a really good screen, what you're really trying to say is the envelope of most of the compounds is noise, so it should be Gaussian. A good screen will actually lead to a tail. So this is actually your number of hits. This is different from the mean. And if you take this screening concentration and someone says, you didn't find hits, screen at a higher concentration. In theory, as you get to higher concentrations, you basically push the mean of the population further. What you really do is you collapse your dynamic signal window, and the hits become compressed up against the upper window. So really, the ideal screen should be where you screen at the lowest concentration possible, where you have a lot of separation and skew and spread of all your assays, so you can detect very strong hits and marginal weak hits. In this case, if I was to spike in actual 20,000 compounds of an IC50 at 10 micromolar, I would expect a bimodal distribution. I belabor this because we're often told after we screen that since we found no hits, to screen at much higher concentrations. And actually, you can argue back that, in fact, you've done a good screen, because I looked at my distribution and I had a lot of skew and kurtosis. Because if a screen gives you nothing but pure Gaussian statistics, Gaussian distribution, that means there are no actual hits, because you won't see any, there's nothing beyond noise. So I think that's important to make. It's just important to tell your management that you've done the screen properly, and there are no hits in that library. So you should find a different library. Final note on reducing false positives and false negatives. Whenever you set a threshold for an assay, if you look at this threshold, let's say there's a region of compounds that you missed. If you set a threshold here, these compounds that you call as hits, if these were all the hits, then if these were all the no hits, then at this threshold, you call, no matter what threshold you set, you have false positives and false negatives. However, if you move the threshold lower, you increase the number of false positives, but you reduce the number of false negatives. But the other way you can do that is by taking duplicates or replicates, because again, that does is shrink the assay variability, and you get more narrow distributions, in which case you can actually reduce the false negative rate while still keeping a relatively low false positive rate. So this is the best way to actually do it. And this to improve data quality and an assay. And this feeds back to, if a Z factor is less than 0.5, you can run replicates. Okay. So just some concluding thoughts. So HTS relies on statistically based rules of thumb, and these are loose statistics. In the case of the Z factor, it represents such a simple rule of thumb that's intuitive and graphically based. It's a quasi-statistical notion of a separation band that accounts for both the dynamic range and the variation of the signal. And it's easy to remember. So all you have to remember is that it's either greater than 0.5 for an excellent assay, and it's between 0.5 and 0 for a doable assay. And so these kinds of rule of thumbs and application to HTS illustrates that, perfect is not necessary if you have something good enough. So it allows you to feel confident in your single point, you actually find bona fide hits. But if you can replicate the assay and do the assay in replicates, then you'll have better signal variation, and that should give you a higher quality screen. Now, in the extremum, you could actually do quantitative high throughput for every compound as a full dose response. And the NCATS and NIH has done a good job of describing the value of that and how you can actually reduce false negatives. And however, most of our rules of thumb really rely and assume that data distributions are normal, which is generally a good case unless you have very extremum values in your data set. Remember, the purpose of high-throughput screening is really just to find initial hits as starting points that represent chemical scaffolds. So rarely do you screen a library of compounds that are all singletons. Each library that I usually screen has multiple families of hits around a scaffold, so it has a mini SAR built in. So the chance of complete miss a scaffold are minimized. And you can mitigate the risk of false positives and negatives using thresholds and looking critically at your assay robustness. So finally, it's important to illustrate that if the hits aren't in a particular library, you really need to screen a different library rather than just simply go back and increase the concentration 2 or 3-fold. So I think that's how I'd like to conclude. So I'd be happy to take questions or comments.
Thank you, TC. That was excellent. I'd like to open the floor for questions. I can start out. TC, do you have any rules of thumb for what would be an excellent Z prime for an assay where instead of single tests, you would go with either concentration response or replicates? I mean, how much lower of a Z prime could you get away with where you would consider it an excellent because you have that additional data for each test agent?
Yeah, I think the Z prime, when Z primes are less than 0.4, I showed that if you do replicates, since this signal standard deviation goes as the square root of the number of replicates, you can improve the data band. So that will then automatically take a 0.4 Z prime assay and make it a higher, like a 0.5 Z prime assay. In terms of what you asked for dose response series, the Z factor itself isn't directly applicable to that question for dose response series. That leads to a different formalism, which I didn't pioneer, but Phil Iverson and people at Lilly have been championing. It's called the minimum significance ratio. And it's more a rule of thumb as if you talk to a chemist and they're in an SAR campaign, they're trying to improve the potency of a compound by a certain factor, a fold. So you like, I want to make it 2-fold better, 3-fold better, 4-fold better. Not so much the absolute value is, but the fold improvement. And so there is a formalism that is related to notions of standard deviations things, but not as simple as the Z prime formula. It leads to checking the assay parameters so that you can calculate how many times you have to run an assay to achieve a significance, a ratio, fold difference in IC50s.
I have a question, TC.
Yes.
So you said a few times that when people come back to you and say, can you please repeat at a higher concentration in the assay? What, in your experience, like, what is the highest concentration you generally do an assay with small molecules?
So that's a loaded question, Sam. So in random libraries, our rule of thumb, I would say, is we really do not feel comfortable going above 25 micromolar. When we start going above that, we actually start noticing data. So what the way it presents is the Z prime controls itself. The assay is robust, but the minute you throw compounds on the Z, the formula, plug-in formula Z rather than Z prime, where you put in the sample means and sample standard deviation, they become significantly graded. Like you might go from a 0.1 0.7 to like 0.4. That means something's really wacky, and you'll show up in the data in terms of scatter is very bad. But more importantly, you'll start seeing non-linearity. Your skew becomes extremely pronounced. And we feel that's not a reflection of the biology, it's a reflection of things like compounds coming out of solution. We'll find very high false positive rates where compounds, when you retest that compound 2 or 3 times, the values just vary all over the place because a lot of these things like precipitation are not linear processes, and they're very time dependent. However, if you're screening a fragment library, which are very small molecules, then many fragment libraries are designed to be soluble up to about 100 micromolar. So that's a very different thing. But my rule of thumb is, when someone asks me to screen at higher than 25 micromolar, as we do for like protein-protein interactions, much higher than that, we feel that you're going to get so much artifacts. So then, not the primary screen, but all the counter screens and all the assay artifact screens become very important. So you can also find out the number of assay artifacts that you have to screen against will go up. So that's a diagnostic library shouldn't be screened at a high concentration.
Thank you.
Do we have other questions?
I can ask one more. Nobody's asking.
Go for it, Sam.
So in your experience, TC, what is the highest factor for a lack of hit confirmation? So I'm asking clearly, I'm going beyond the screen now.
I can tell
Most
you
common cause. Yeah, go ahead.
Well, here's the diagnostic. If you have an assay, let's say it has a Z factor, Z prime factor of the pure assay of like, 0.8, and then you throw a library against it, and you still like maybe 0.75, then you call hits and you retest them, and none of them confirm. Something's really screwy there. That's not actually, I think, a function of the statistics. Something's happening with it could be that the assay is set up in a way that's at non-perturbable ways. I think one of the comments that Barnes Zweibach made was a reliance on much higher. For many assay developers who are trying to get very high Z primes, you are setting assays to be pegged, so they're really like maxed out in terms of the signal window. So it's kind of like saying you're screening at 10 km instead of 1 km of an enzyme concentration. So what you really have to do is really have to have a compound that really inhibits that and can compete. Let's say it's a kinase and you had high ATP, then your compound, if it's an ATP competitive inhibitor, might never be seen, right? So that's where they worry that an over-reliance on high Z primes makes people blind to the limitations of their assay. In the case where the assay statistics are really tight like that, you screen, you'll find no hits. That means you over-specified the assay. If you find hits, but they're all don't repeat, I think that's something else. It means that what you're interrogating is not the biology. You're interrogating some physicochemical property of the compound. And you can often check, when we talk about irreversible inhibitors, remember, so you might, an assay might have taken a while by the time you analyze that point, but when you redo the assay, you're at the earlier time point because you can't read a plate instantaneously. It takes a couple minutes to go across a plate. That's the best I can do for that.
Yeah. Thank you.
As I was preparing this talk and I looked at those, especially the more recent paper by D.H. Zhang, very complex statistics. I mean, I agree with some of what they say. It's true. I have to emphasize that what we appealed to was, and why people got excited was, it's a very simple graphical formalism of two noise envelopes touching each other or being separated from each other to give you a zone of separation. I think that's what made it intuitively appealing and easy to remember. And it is not formal, and you could argue it's not proper use of statistics other than saying that the standard deviation error band is normally distributed. Having said all that, it's been 1999, right? And it's been holding steady until this 2023. There's, new papers challenging that, but I don't disagree with what their conclusion is. I find it hard to apply the new papers in a day-to-day without having a statistician by my side.
Do we have any other questions for TC?
I'll get a lot of statistician hate mail, that's for sure.
Thanks very much.
Okay.
Take care, everyone.
Thank you.
Goodbye.